So I think we can get started now. So we already have 46 people that are joining in. So that's a great sign. I was hopeful that we'd have a lot of interest, and it seems like we definitely do. So I'll just introduce the talk first, and then I'll turn it over to Dr. Ellis. So welcome. This is the first webinar that we're having. It came about through a conversation between some faculty in the arts and sciences who gave us some feedback about how actually to set this webinar up. And the idea is, well, it came about for a lot of reasons. I think there's a lot of interest amongst students, staff, faculty in learning more about this pandemic, this disease, this virus. And fortunately, in the arts and sciences and throughout the school, we have a lot of people that are really well informed about these issues. So we thought it'd be a great opportunity to bring faculty in so we can have these sort of engaging conversations. Also because we've all been, most of us at least, have been stuck at home for going on, 4 months now, 3 months now, that I think a lot of us are looking for some opportunity maybe to engage with each other. I mean, still digitally, obviously, but some way of doing that. And just to highlight the expertise that we have here and to get sort of a conversation going. So just a brief introduction, Dr. Ellis is a professor in the biology program here. She's an expert in virology. So I thought, what better person to lead off these talks than somebody that knows about viruses? Because fundamentally, that's what we're talking about. This is what started everything. So now I'll turn it over to Dr. Ellis now.
Thank you. So thanks for coming to this first session of the webinar series. So I'm going to focus on a brief introduction to COVID-19, the disease and the spread, but that's going to be fairly brief compared to the time I'm going to spend talking about the biology of the organism and what we learned from the genomic studies of the organism also. And so this is sort of part of a larger series I created for the first year biology students in the spring, where I created 5 different videos all talking about different aspects of the pandemic. And so the first was an introduction, the second was about the virology, and that's for the most part what I'm going to be talking about today. I talked a lot about, in that one, the pathology of the disease, the epidemiology, so who gets it, how it spread, in the origin, and then I talked a little bit about the science of social distancing and some of the treatments that are being explored right now. So this is a much shorter version of that. So I do talk a little bit about, introduce it a little, but I wanted to focus a little bit more on what we're able to learn from genomic studies and other types of research through COVID. And so I've prepared some slides. So what I'm going to do is I'm going to share my screen so you guys can see the slides as I'm talking. And so hold on, let me try to figure that out. Okay, all right, so as mentioned, Dr. Crystal Ellis. I'm an assistant professor of biology at MCPHS University. I teach genetics, biochemistry, general biology, a few others, and I'm currently teaching microbiology over the 10-week summer session right now. I also do research in the area of cholera biology, which focuses on how the bacteria Vibrio cholera adapts to become tolerant to low levels of chlorine. So I've always had an interest in microbiology, and I wanted to join this webinar series to provide information about COVID, since I've done kind of a deep dive already for my biology course in the spring, but also to allow people to ask me questions about COVID. Again, I'm not an expert on COVID, but I am a microbiologist, so I do have some expertise on this. But first, I wanted to point out that all of this information that I'm about to talk about was gathered using these references. And there are others that I used to create the other 4 parts to the 5-part series that I created for my bio class. So I tried to provide links for all of these sources in case you get curious and you want to do kind of the same deep dive that I did. But it is important to recognize that information literacy right now is even more important than it's ever been. It's really critical to make sure that you're reading bits of information and news reports with a skeptical lens and a critical eye. Things change really quickly and daily because of this pandemic, and it's really hard to keep up with all the information as it comes out. You know, how to sort of critically evaluate the news sources they're looking at. And so if you're looking at a news source and they're referencing something else that you can't sort of find the original study or original content or data to sort of support the claims that you're reading about, if it were me, I would give that information less weight. If you aren't able to find multiple sources for that claim or that finding, or if it weren't put through the peer review process, I might not let it I might not choose to let it affect my daily decisions about how to manage the pandemic. So just be careful about what you're reading, and just always use your most critical eye when reviewing information that you want to use to help inform your choices or help inform you how to stay safe or how to help inform others to stay safe. So just use a critical eye when you're looking through information. Okay, all right. So the first thing I wanted to do was introduce some terminology because there are three terms that are sort of thrown around and used interchangeably here. And they mean kind of different things. And so what is coronavirus when people are talking about that? Coronaviruses are a group of viruses that typically cause mild symptoms and mild illnesses like the common cold, but others can cause really severe viral pneumonia that can infect the lungs and lead to things like pneumonia or bronchitis. Most people do become infected with coronaviruses at some point in their lives, but the majority of those illnesses are harmless and don't really lead to hospitalizations or severe fevers or things like that. However, there are a few that do, and SARS-CoV-2 is one of them. So this is the virus that causes COVID-19. COVID-19, of course, referring to the disease that is caused by the virus. SARS-CoV-2 is the 7th coronavirus known to infect human beings. SARS-CoV was the virus that caused SARS, and MERS-CoV was the virus that caused MERS, both of them coronavirus. And then all three of these guys can cause severe disease in the form of a viral pneumonia. So when COVID first emerged in Wuhan City in the Hubei province of China in December 2019, the Chinese government sort of started warning of the spread of this virus through their communities that was landing people in the hospital, especially people over 60 years old. And by January 22nd, there were 580 cases. 2 days later, that number jumped to 1,300. And so this is an exponential growth of case increase. And the reported ease of spread from the doctors on the front lines caused the World Health Organization to raise alarms and declare the spread of COVID-19 a public health emergency on January 30th. As international travel continued, testing started to ramp up and the virus sort of started to spread quickly throughout the globe. And on March 11th, only a few months later, about 3 months later, the WHO declared COVID-19 to be a global pandemic. So we went from a case detection in China in December 2019 to a full-blown pandemic by March 2020, only 3 months later. And nearly every country and territory is or was affected by COVID-19. So how did this happen? What happened? And there are, of course, many contributing factors for fluctuations in the spread of a disease, the mortality of an illness, or how many cases a country gets from one country to the next. But there are a few features of the disease itself and the virus itself that resulted in the fast spread of this. So first and foremost, we are an immunologically naive population. This means that we have little to no natural immunity to prevent infection of this virus. There's no baseline immunity, no natural defense that we have to combat this. For illnesses like the flu, only some people are susceptible each year, not every single person on the globe. And this is because, say the major circulating strain this year was like a minor circulating strain last year. Some people still got it last year. And so therefore this year they would have been recovered and are immune to it this year. So exposure to related strains in previous years can sometimes provide for cross protection against the flu. There's a vaccine that continued vaccination with these vaccines will help give you more and more protection against flus in coming years. But this isn't like that. This is a brand new virus. We don't really have any previous exposure, no exposure to any highly related strains, no vaccines, nothing like that. So everyone is potentially infectable to varying severities or degrees, but everyone is potentially infectable. So this explains these sort of terrifying numbers that we keep seeing increase globally. So I looked up the total numbers on June 12th, so last Friday, I think it was. We had 7.5 million confirmed cases in the globe and 423,000 confirmed fatalities as of last Friday. In the US alone, which leads the pack, which is not really a race we want to win, but we are very much winning it, there have been 2.1 million confirmed cases in the US and 118,000 confirmed fatalities as of Monday of this week. That's 29% of the confirmed COVID cases in the world are just in the US. 28% of the fatalities in the world are just in the US. So we're not handling this great. We're not doing a great job. You know, there are pockets where we're working really hard. Obviously, people are working really hard, but we are leading the pack for the most cases and the most fatalities. Excuse me. So second, COVID spreads really, really fast through very casual contact. And so transmission is primarily through this respiratory droplet. This is not to be confused with aerosol droplet. Respiratory droplets are bigger and they don't linger in the air for as long as aerosol droplets do. So this is a respiratory droplet transmission. And so these are, this means the virus is suspended in the little droplets that exit your mouth and your nose when you breathe, cough, or talk. And they float around and can float through the air for about a distance of 6 feet, 3 to 6 feet, which is the rationale behind the 6-foot distance rule when you're social distancing. There are some reports about it being spread through contact with contaminated surfaces, although the CDC did release a paper, I think 2 or 3 weeks ago, describing that the primary route is really more respiratory droplet and that it's actually harder to get from a surface than we previously thought. I'm not saying you should stop washing your hands. I'm definitely not saying that. But it's a little bit harder to get it from a contaminated surface than we originally thought. So you don't have to like bleach wipe all your apples, but I would still, you know, wash your hands and you know, practice hand sanitation and hygiene. The best estimation of how a disease spreads is the reproductive number, or the R0. And this is an estimation of if somebody becomes ill with a sickness, how many people they're likely to spread it to while they're still sick. And so for the flu, this is 1.5. And so what that means is if you get the flu, you are potentially going to infect 1 or 2 other people while you're sick. For COVID, it's about 2 to 4. So it's really hard to measure since we're still in the middle of this pandemic, and things like social distancing and other control measures can affect these numbers a little bit. But in the earlier days when we weren't really doing social distancing as quickly, so right when this all started, the best estimate was about 2 to 4. And so that means everybody, any person who has COVID-19 can potentially infect 2 to 4 other people. So this spreads really fast. And then lastly, the case fatality rate for COVID is not trivial. As of today, COVID-19 is more deadly than the flu. It is hard to make this comparison, and it is a tough thing to measure. Mortality is affected by so many things. Testing variabilities, so how many people get tested really will affect mortality rates. People who are being tested right now are people that are ill and are showing symptoms, but we know that there are people that have mild symptoms or are asymptomatic and still infected and able to spread the illness. And so those people might not come to the hospital or get tested, and so those aren't incorporated into the statistic. And so as testing sort of ramps up, and as we do retrospective testing, which is like antibody testing, so we can figure out how many people were sick and maybe didn't realize it, mortality rates are going to change based on, you know, how much we're testing people and who we're testing. It also depends on access to healthcare and whether or not the healthcare system is overburdened, and that can affect the mortality rates too. And so more people will die from COVID if they can't get the medical attention they need to survive, if the healthcare system is overburdened, overrun, or not working properly. And so people who would otherwise survive COVID might be dying because they don't have a healthcare system they can take advantage of to get them well. And so the simple math for the death rate for COVID is about 5% in the US compared to the flu, which is 0.1%. So it is at least today more deadly than the flu. And the death rate globally is about 6.4. And this of course varies from region to region based on access to healthcare and things like that. I did look at a couple of published studies to kind of see what the studies were saying was the mortality rate. And again, depends on the population you're studying. It's sort of all over the place. In a Chinese analysis of 72,000 cases, which is the data you're seeing here on the right, 2.3% of those confirmed or suspected to have the virus died. And so the mortality rate is 2.3% being reported from that analysis. However, in an Italian study where the proportion of residents over 65 is much higher, the fatality rate was measured at about 10% as of March 25th. And so again, it really does fluctuate depending on where you're looking. Let's look in the US since that's where we are. The estimated mortality rate in the US for people aged 65 to 84 is anywhere from 3 to 11% depending on the age group. But below 64, so any ages 55 to 64, it's about 1 to 3%, and then those younger than 54, it's 1% or less. And so it really does sort of showcase who the high risk where the high risk groups are. But regardless of how you calculate mortality rate, the deaths due to COVID-19 in raw numbers tell a pretty powerful message. And so this was an animation that I borrowed from a Reddit channel. So this is not like published data or anything, but these are raw numbers of COVID. And what this is showing you is the number of COVID-19 deaths versus other epidemics since 2000, from the day of the first death. And so this carries out over 100 days. And you can see cholera up there leading the pack for the number of deaths globally in the world. But here comes COVID on that orange bar. And this is basically showing you, like, there's so many numbers of people who are being affected by this. This is why this is a serious public health disaster. This is why the WHO declared a global public health emergency, with many individual countries declaring national public health emergencies. This animation stops at 100 days when COVID reaches about 88,000 deaths. But just to sort of show you, cholera, which isn't anymore, was the only real contender for something like this. Cholera kills about 200,000 people every year. COVID-19 has already killed twice that amount of people in half that amount of time. And so this is a huge problem that we need to focus on and care about. So this thing is killing lots of people, bringing misery to all of us in different ways, be they economic burdens or health burdens, burdens to our ability to care for the sick, care for the elderly or the very young. And what really seems unfair about all of this, totally unfair, is that this is a bit of RNA wrapped in a little bit of protein that's bringing down our big, big brains in our giant societies. Actually, the name of my talk was borrowed from my aunt, who is, she does work in healthcare, and she said to me in a text, "Isn't it amazing how just a little bit of RNA and a little bit of protein can turn our world upside down?" And I thought that was such a powerful statement coming from somebody who's on the front lines. So let's talk a little bit more about the biology of this thing. So I'm going to talk a little bit about viruses in general, and then I'm going to talk, connect it specifically to SARS-CoV-2. And so viruses in general are essentially a protein outside and a nucleic acid inside. That nucleic acid can be DNA or RNA, and that genome is responsible for creating the proteins that make up the outside of the particle. Some, but not all, viruses are enclosed in something called an envelope, and this is made up of lipids or fats arranged like a membrane, the same kind of membrane that encloses all of our cells. And the viruses can steal our cellular membranes in order to make this envelope in some cases. So this is how they hide from our immune system, is they throw on a little coat that looks just like our cells. And so our immune cells don't necessarily flag them as something to be worried about. Some viruses also have spikes. These are a combination of carbs and proteins that stick out along the outside of the virus. That's what these little guys are. Most viruses only infect specific types of cells in a single host, but changing a host is possible through adaptations in these attachment sites specifically. Some attach very strongly, and this would be the virus's main host, but other times the connection between the viral spikes and their target can be kind of weaker. And so this would be able to maybe live for a moment in that person or that host, or for a short period of time, but maybe not cause severe disease. But this is how host range can actually be bigger than just a single organism, but not nearly as big as like every organism. There are other factors that contribute to host range, not just the spike proteins. Viruses are classified based on what their nucleic acids insides are, the shape of their protein outsides, and whether or not they do have an envelope. The protein outside is called a capsid, and the shape, again, like I said, is determined by the code in their DNA or their RNA. The spikes also are determined by that code, and they change often and are subject to evolution by natural selection. And I'm going to come back to that when I talk about the origin of where this thing came from. The proteins on the spikes, those spike proteins mutate very often. They do in the flu too. So this is why you can be infected by one strain of the flu, recover, and have antibodies that attack that strain. But if those antibodies are made to the spike protein and that spike protein changes in the following year, your antibodies will no longer protect you. And so you're open to infection again. That virus can reproduce and spread in your body. Coronaviruses, in general, are very large RNA viruses. They have a genome about 26,000 to 32,000 base pairs of RNA. That's pretty big compared to the flu, which is also an RNA virus. The flu is 13 to 14,000 base pairs, so about half as big. SARS-CoV-2 particles, excuse me, are surrounded by an envelope, so they do have that fatty outer layer, and they usually appear like tiny spheres with little spikes hanging off of it under high-powered microscopes. Those little spike proteins arrange themselves in group of 3 and look like a little crown, and so that's where coronaviruses get their name, corona meaning crown. These viruses bind to intestinal epithelial cells, spleen epithelial cells, and also lung epithelial cells. Most importantly, one of the receptors that the spikes bind to for SARS-CoV-2 is the ACE2 receptor on lung epithelial cells, and this is how they're able to cause a respiratory infection. Okay, so let's talk a little bit about the life cycle, what the virus is doing once it enters your body. This image is taken from a review on the SARS-CoV life cycle. So this is the virus that causes SARS. So this is not specific to SARS-CoV-2, but it's pretty close, if not the same, how SARS-CoV-2 reproduces in the body. So there are 6 major steps. The first is attachment, and that's this interaction between the spike protein and its target receptor. And in this case, it's receptor ACE2. So SARS-CoV attaches to the same receptor that SARS-CoV-2 does. They both attach to the ACE2 receptor in lung tissue. Once the attachment is solid, the next step is penetration. And the cell sort of reaches out and engulfs the virus, takes it into its own cell in a structure called an endosome. So that's what this is. It's sort of like the host cell takes a little bit of its own plasma membrane and wraps it around the virus and pulls it into the cell. Next is the uncoating step, which is the process of releasing the viral contents into the cell. And there's lots of ways to do this. There's lots of cellular factors and viral factors that contribute to this process. But essentially, the envelope of the endosome, the envelope of the virus fuses with the membrane of the endosome, and then all the insides spill out. So the protein capsid spills out, and then that will dissolve, and the genome spills out. And so now the viral genome is floating around in your cell. And the next step is biosynthesis, where the viral genome is copied and made into proteins by your own host cell machinery. And so what your cells use to copy your own genome and make your own proteins is stolen by this virus, hijacked to replicate its own genome and make its own proteins. The SARS-CoV-2 genome is one long single piece of RNA, and the very first step in this process is to take some of that, so your host cell machinery will copy some of that into a protein called replicase, which will chop up the rest of the genome into smaller pieces. And it's not destroying it, it's chopping it up to sort of process it and get it ready for making these different proteins that are listed here. So each of these will create one, and in some cases, 2 different proteins. All those proteins start to sort of float around, and then some of them are actually responsible for directing the next step of the life cycle, which is assembly. During this step, viral proteins will come together and sort of make up the outside, the protein outside. There's other things that are happening too with these proteins that I'll talk about in a minute. But essentially, we have the virus is working to assemble itself, basically build the protein outside, stuff it full of RNA insides, and then wrap that lipid coat around it again. The final step is release, where the viral proteins, again, are recoded by the envelope, and they exit the cell sort of the same way they came in by this endosome process. But as they exit the cell, they can poke holes in it, they can trigger inflammation, they can trigger cell death. And so that's what leads to the cell death, as that's part of the pathology of the disease. Okay, so what's in that genome? There was a sample of SARS-CoV-2 isolated from a 41-year-old man who worked at the seafood market in Wuhan, where the first cluster of cases first appeared. And so 27 of the first 41 cases were linked to the seafood market in Wuhan. And so this, the sequencing of this virus represents like the earliest that we could get to with the patient 0. This genome was fully sequenced, and many research groups have subsequently analyzed the sequence information, trying to help us understand how the viruses hijack immune cells and your body cells, how it leads to severe illness, what what features of the virus can cause a severe illness that's different from other coronaviruses that are so mild. It also helps us to confirm the origin of the strain, which I'm going to discuss on its own in a moment, and other things like that. So what we've learned so far is that this genome contains information for 29 predicted proteins. And so some of these proteins, we don't know what they do. We can sort of make a good guess based on comparing them to similar proteins in other viruses, but there are still some unknowns as to what some of these proteins are doing. But a lot of them, we do know what they do. Some of them help to make copies of the virus during the biosynthesis step. Some of them help to suppress the body's immune responses, so suppress your own body's way of controlling viral spread. And there are several ways in which your own body will try to do this. SARS-CoV-2 Genome makes proteins that sort of interrupt that process. There are some proteins that slow down, specifically slow down the infected cell's production of its own proteins. And so there's more materials and energy left to replicate the viral proteins. And this also puts a stop to your own host defense mechanisms. We do have antiviral proteins that seek out and find viral RNA in our cells and break it up into small bits so that it inactivates it. But there is a protein made by this virus that sort of camouflages the RNAs, the virus genes, so they can't get attacked as easily. These proteins make up the spike protein where they attach. The proteins also are involved in allowing viruses to escape. And so those ones would be important for mounting the inflammatory response and the cell death response. And so we can learn a lot from this information. And knowing more about the viral biology and the viral genome helps us to combat the pandemic. So continued research on the viral structure and the genome is really, really important for us to try to control and use the proper infection methods to control the pandemic. Knowing that the virus spreads through very casual contact helped us to determine that face coverings and hand washing measures would go a long way to reduce the spread of the virus. By studying the structure of the virus, we learned that it has an envelope. And so things like alcohols are really effective disinfectants against things that have an envelope. Those alcohols break up the lipids. And so the virus can't attach or penetrate the cell if it doesn't have that envelope anymore. Not all viruses are like this. For example, norovirus, which is sort of like I hesitate to call it food poisoning, but you can get it from contaminated food. Norovirus is like a 24-hour stomach bug that causes like severe diarrhea and vomiting. Everybody's had it. It's like that horrible 24 to 48 hours where you're just miserable, throwing up and having diarrhea all day long. Those viruses don't have an envelope. And so things like hand sanitizer will not kill them. And so if someone in your household has the stomach bug, has this kind of thing, and it's norovirus, and you think you're super smart by using hand sanitizer to wash your hands and you're just like bathing in hand sanitizer, you might get sick anyway because they're not super effective against norovirus. But with SARS-CoV-2, hand sanitizer actually works pretty great because it destroys the viral envelope. And so again, use of a hand sanitizer along with face coverings and regular hand washing with soap and water are great infection control measures. And so knowing more about the viral structure helped us to figure that out. Knowing more about the genome also helps us with things like drug development, vaccine development, determining the origin of the strain, predicting the likelihood of future pandemics, or for this one becoming more severe. In terms of drug development, many viral proteins can be the target for effective drugs. So for example, we can develop drugs that interfere with the spike proteins that help with attachment. And so if those drugs interfere with the spike protein, the virus can attach and they can infect a cell. If they can't infect a cell, they can't complete their life cycle. And so that would stop the infection. There are drugs that can be developed to target other parts of the viral life cycle, like the uncoding process. This is actually what chloroquine does, one of the drugs being tested for COVID treatments. The first step so that's just one of them. Remdesivir is another potential treatment. This interferes with copying of the viral genome, so other parts of the biosynthesis step. And so learning more about the types of proteins that are present helps us to develop drugs that target them. And of course, are potential vaccine candidates for developing antibodies against. Genome analysis also helps us to confirm the origin of the virus and track relatedness to other coronaviruses and related parent strains in other animal hosts. So I'm going to come back to this point. That's the point I'm going to end on. And then still other genomic analyses help us to predict the likelihood that this will happen again or become more deadly by tracking evolution rates between related strains or mutation rates of this current strain and other related coronaviruses. Current reports do show that this strain mutates slowly, and so this reduces the chance of it mutating and becoming more deadly. It doesn't obliterate it, it just reduces that chance. However, there is strong evidence to show that SARS-CoV-2 transmitted from an animal host to a human host, very much like its other coronavirus buddies like SARS and MERS viruses. So the likelihood of this happening again is not unlikely. It could potentially happen again. And so what we can learn from this one will help arm us for the next one. And so practices that led to the interaction between host animals and humans, if we can understand more about how this happened, then we can put control measures in place now to prevent it from happening in the future. And so this brings me to my final point, which concerns the origin of SARS-CoV-2. Genetic research about COVID in a field of biology called comparative genomics can help us to dispel rumors about things like viruses being engineered in the lab and intentionally released into the public and things like that. So let's dig into this a bit deeper. And I'll talk about the evidence that supports a zoonotic transmission of COVID and why this evidence does not support genetic engineering of the virus in a lab. Zoonosis, or zoonotic transmission, means that the disease is transmitted from other animals to humans through the air. So this was the case with swine flu, or through bites and saliva, like when rabies is transmitted from an infected animal to a person, they do that by biting. Zoonotic transmission is really common for coronaviruses and has been the link or the origin of other coronavirus outbreaks. So SARS jumped from civets to humans, and MERS jumped from camels to humans. And both of those had an original bat reservoir where they were first in bats and then somehow jumped to civets and then to us, or they were in bats and they jumped to camels and then jumped to us. And so this is a fairly common mode of transmission for coronaviruses. I've read through several papers, all of which are referenced on the slide that I showed at the beginning, and I've read the analyses and they all support that SARS-CoV-2 is transmitted by an animal host. These studies, again, use something that I mentioned before called comparative genomics, where they compare the sequences of SARS-CoV-2 and the functions of the proteins in SARS-CoV-2 across multiple coronavirus genomes. So SARS, they compared it to SARS virus, the MERS virus, and then other coronaviruses isolated from bats. And there are two notable studies that I found. One is related to the whole genome comparison, so the entire genome of SARS-CoV-2 compared to others, and then also just looking at the spike proteins genetic material compared to others. And so first I'm going to talk about the whole genome comparisons. So first, the SARS-CoV-2 RNA sequence was compared to the sequence of SARS-CoV, so the virus that causes SARS, and also MERS-CoV, the virus that causes MERS, and then other SARS-like coronaviruses found in bats, or reported to have been found in bats. They did what's called a phylogenetic analysis, which is useful for predicting how related genomes are to each other. So lining them up and looking at the differences between them and measuring them helps us to understand how related the SARS-CoV-2 virus is to these other ones. And it turns out that SARS-CoV-2 virus is more similar to the SARS-like bat coronaviruses than either the SARS or MERS viruses that were sort of wreaking havoc in the human population before. In another study that examined a sample of COVID viruses isolated from patients, and then they isolated two SARS-like bat coronaviruses from bats. So they took COVID viruses from people, and then they took SARS-like coronaviruses from bats. In Eastern China, they isolated these after the pandemic started. And it turns out that the SARS-CoV-2 viruses are about 88% genetically identical to the two SARS-like bat coronaviruses. Whereas if you compare it to the SARS virus, it's only 79% similar, and really only 50% similar to the MERS virus. And so it's genetically the most similar to viruses being pulled out of bats from the wild. Another study found that 27 of the first 41 reported cases were linked to one seafood market in Wuhan, where there are trade of animals, and so this represents an opportunity for a jump to occur from infected animals to people, since there's lots of contact in that situation. So that's the first piece of evidence, that it's actually most similar to strains in bats. And so if people were going to try to engineer a virus, they wanted to infect people, why would they choose a basis or like a base viral genome that's coming from a bat that has no known connection to human disease? They would probably more likely choose something that was already known to cause illness, like tinker with the SARS virus or tinker with the MERS virus to get this to happen. But that's not the case. So it doesn't logically make sense that somebody who wanted to engineer this virus would go to a bat virus genomes or bat viruses and start there, because there's no known really known way to know that those would actually cause human disease. So let's consider another point. This is about the spike protein. Remember that this is the part of the virus that tends to mutate often, and it's responsible for host specificity. So it determines if the virus can attach and enter a cell. When comparing the genomes of several coronaviruses, there are mutations in the receptor binding, so in the spike protein, that are distinct from other coronaviruses. They are the most similar to the SARS-CoV, the SARS virus, which in a previous slide you saw that it was able to, SARS-CoV, the SARS virus, is actually able to attach to the same receptor. But there are 6 amino acids on the SARS-CoV-2 spike that are known to be critical binding spots for ACE2. So we knew that this is where mutations would likely happen, and that's where they were happening in SARS-CoV-2. And so these kind of spikes, these SARS-CoV-2 spikes, seem to have high affinity to the ACE2 receptors in humans and some other species too, like ferrets and cats. So experiments with the virus itself reveal that those 6 amino acids allow it to bind very tightly to ACE2, but in a separate study where they used computational analysis to predict how that mutation would affect binding to ACE2, the computational analysis predicted that it doesn't actually bind well. And so what really is happening is that those spike proteins, those mutations in that area, make it so that the virus can stick really well to ACE2. But if you take those same mutations and subject them to a computational prediction software, the computer predicts that it's actually a terrible interaction. It doesn't actually bind well, and it predicts that the SARS virus binds better. And so again, if somebody were to try to engineer a virus in the lab, why would they choose a set of mutations that computationally predicts that it wouldn't work? I mean, why would they do that? That doesn't logically make any sense. And so these two things kind of provide strong evidence that SARS-CoV-2 is not the product of purposeful manipulation, but rather they're the product of evolution by natural selection. We would have predicted that these mutations didn't work to bind very well, but in fact, in reality, they do bind really well. And so there could be other factors, other cellular factors that we're unable to predict that allowed for that binding to happen. So again, if someone were trying to create a virus in the lab that they wanted to bind to ACE2, they would have likely copied mutations from other coronaviruses known to bind well, not mutations that are predicted to be terrible at binding human receptors. And so one study proposed 2 potential scenarios wherein the virus was entered the human population via zoonotic transmission through the process of natural selection. So the first scenario is, the researchers suggest that SARS-CoV-2 originated in a bat, and that bat transmitted the virus to potentially another intermediate animal, or directly. And so it's not totally known yet if there's an intermediate host or if this was direct contact between a human and a bat. More research is necessary for that. But in this scenario, basically what happened is that the genetic features that make this new coronavirus so effective in infecting humans, so the mutations in that spike protein, would have happened before hopping into humans. And so this mutation wasn't really doing anything significant in the bat, but once there was contact between the bat and the person, the virus jumped and suddenly found itself in an environment where that mutation was extremely beneficial. And so then it would reproduce, thrive, and then flourish. The second scenario is that the virus jumped from bat to human before it evolved its ability to infect humans really well, so before that mutation occurred, and then prolonged living inside of human hosts and human populations. Eventually that mutation rose up, and then if that mutation allows for better binding, then that will be passed on to the next generation in higher numbers, and then would favor that mutation. And it's likely that that mutation allowed it also to spread from person to person. Determining which scenario is the answer requires a lot more research, but it could help predict the future of this pandemic. So if the virus did enter human cells already able to infect humans, this raises the possibility that future outbreaks will happen. But if the virus had to first jump into humans, kind of get used to that environment, and then evolve to become infectious, that's less likely, right? Because that's two things that sort of have to happen. And so that reduces the chance that a future outbreak would be possible. And so these kinds of genomic studies really do help us with lots of different things. They can dispel rumors about things like the origin wasn't engineered in a lab. It was zoonotic transmission. And then also it helps us to predict the outcome of this. Is this going to happen again? And where is it likely to happen? And that will help instruct us on ways and proper measures to prevent this from happening again. That's all for me. All right, so any questions from the group or anything you want to discuss, like go nuts, ask your questions.
Yeah, we had a few questions that popped up in the chat already. So Kim was actually asking about, I think it's probably prompted by the fact that we've seen a lot of racial disparities in cases and deaths from COVID-19. Whether, to what extent that might have affected some of the statistics that you were presenting. I didn't know if you had any thoughts about that.
Yeah, I mean, oh gosh, for the mortality rate, so many different things can affect the mortality rate and how we measure it. The biggest one being we're still in the middle of this thing. And so it's really, mortality rates are not easily measured when you're in the middle of a pandemic. It's really not until later that we can get a full picture of what was going on when it was going on. But certainly that could have an effect. So for example, there's lots of evidence to suggest that people in different socioeconomic statuses are unable to get access to healthcare that they need. And that has sort of set the stage for pre-existing conditions in certain groups of people because they weren't able to get the help they needed before the pandemic happened. And then when it happened, they had pre-existing conditions on top of still not being able to get the care that they need. So certainly that does account for the disparities you see in mortality rates from, even community to community within a single state, or from state to state, or from country to country, for sure. Absolutely, that's definitely one of them.
Great. And then Elizaveta Burson asked, is it possible that COVID-19 started spreading earlier, but doctors did not realize that because the symptoms are similar to the common cold? Really interesting question.
So that is certainly possible. Again, we don't know who's affected until we can start testing. And so if you aren't testing, then how can you know how many people are actually ill? And so we do suspect, some studies, again, I don't know, I can't think of like exactly what the source was because I've read a lot of things, but I have read that it's possible that this virus was circulating before our ability to test for it happened. And so the spread had already started. And because the majority of cases are mild or moderate enough to recover at home, it's possible that it was circulating and we just didn't know, and that there was an uptick in pneumonia-related cases during a flu season that was already ongoing in our country. And so it might not have raised any flags like, what is this weird pneumonia that people keep getting that isn't the flu but is something else? We don't know. And then it wasn't really until retroactively that people started to realize, oh, that could have been COVID that was harming those people. In fact, my own pediatrician, when I called her to ask about my son, like what is the likelihood of him catching this illness or things like that, she said, I have been seeing illnesses in my office that wasn't strep, that wasn't flu, and I just didn't know what it was. And it's totally possible that it could have been COVID. And so, yeah, I would say before we started testing, it's incredibly possible that there was already spread in our communities before we even knew what to test for.
Yeah, there was a study, I think it was from France, and they were looking at patients who had died from respiratory infections, and they going back to November, and they were saying some of them were presenting similar clinically to COVID-19, but they didn't, they weren't testing for the virus itself, so it could have just been similar.
Yeah.
Virology.
And it is tricky because it's a viral pneumonia. Viral pneumonias are caused by lots of different types of viruses, not just COVID. This cytokine storm that everybody is talking about, which is the over aggressive immune response that you get to the virus, is common in flu complications also. And so the one thing that does seem to be fairly unique to COVID is what's called a bilateral viral pneumonia, meaning both lungs are infected, not just one. So it's typical for a viral pneumonia for only a single lung to be infected and for that to be the cause of the problem. But for COVID specifically, they're seeing a lot more bilateral pneumonias. And so there's lots of there's an RNA test that tests whether or not you have the infection actively. There's an antibody test that shows that you had been infected and have since recovered. And then there's another test that's not as frequently used, but it can be, which is a chest x-ray, looking to see what kind of pneumonia you have. And if you have a bilateral pneumonia, that's really suspect that it could be COVID. That's like a hallmark feature of COVID-19.
Jen was asking about how to critically review literature that's published about COVID.
Things are being published at record rates right now. So the peer review process is a little bit compromised because people are trying to get information out there as fast as possible. But yeah, you do have to be critical. So that report that I mentioned about the CDC point out this report that said, oh, it's less likely to be transmitted on contaminated surfaces. That is not blanket advice to say, oh, just stop washing your hands. It doesn't really matter. No, definitely keep washing your hands. So like I said, when I read that, I was like, oh, my Amazon packages are probably not teeming with COVID. I can probably stop bleach wiping all my Amazon packages. But I am going to keep using hand sanitizer at the gas pumps. I am going to keep using them at the grocery store because, it's not unlikely, it's just less likely. And so you can sort of gauge your response there. You don't have to read everything and take it all as gospel, because everything is happening really quickly. Information is changing daily. And so try your best to turn on those information literacy skills and look for, sources as best you can. When that peer review process kind of buckles a little bit under the pressure, you haven't just accepted it as gospel and changed all of your lifestyles. You incorporated it into what you're already doing. And so measured responses, guys, that's what you really want to kind of go forward with this, is have a measured response to this. So read everything as much as you can, but again, critically evaluate the sources of where these things are coming from and make adjustments as changes come up.
I'm going to combine two questions. So one coming from Dr. Deanne Ho and then also Rachel Carney. So Deanne basically asked about there being some evidence that some people that are affected with the virus don't have an antigenic response, which I didn't actually know about, which is interesting. And he was wondering about how that might affect the development of the vaccine. And then Rachel was just asking about how confident you are about the development of a vaccine, which I think everybody's wondering about, right?
Yeah, well, okay. It is true. I have read those reports also, that there are some people who seemingly do not have an antigenic response to COVID. What that means, sort of demystifying that language, is you get COVID, you probably have an antibody response that protects you initially, but those antibodies sort of disappear. That has been reported in some places. I have not seen it reported in all places, and I've seen the opposite report also. So I think it's very much a person-to-person situation. How good is your immune system at mounting a response in general that's going to affect it? I've studied this a lot from the lens of cholera also. So for example, when people get cholera and survive, they're protected from getting it again for 10 years. They won't get it again, even if challenged. The vaccine against cholera that we currently have will protect you for a year or two at best. And so that's very bizarre to people who are trying to develop the vaccine. What's in the cholera vaccine is dead cholera. So all the same bits and pieces that your body would be exposed to are in the vaccine. But for some reason, there is a big difference between something that is dead and something that is alive in mounting an immune response. Why? We don't know. That's why we're researching it. We honestly don't know. And so it could be a similar situation where, we're trying to put little bits and pieces of the COVID virus in the vaccine to try to mount an immune response, some of those bits might work better than others, or it might not work at all. And we don't really know what the outcome is going to be. The good news is there's lots and lots and lots of vaccine trials going on. There's lots of candidates, and there's lots of people working on this problem. And so I'm trying to stay positive in the sense that I'm hoping that maybe 1 or 2 vaccines of the 60 will work. And so we have a little bit of option. But I find it hard to believe that all of the researchers that are currently on this problem will make no progress. That doesn't make any sense to me that that would be the case. I guess I'm not relying on a vaccine to get us out of this mess, especially now, since the vaccine itself, even if one does work and it works well, we're not likely to see it for a while. And so what are we going to do now? And so that's why my shift has been towards social distancing and trying those infection measures to keep us safe, and just giving those researchers the time they need to develop something, whether it be a drug for treatment or a vaccine for protection. But I honestly don't know. There have been the method being used to develop the vaccine that's sort of the furthest along hasn't really worked very well in other systems. So it's not, but so far the data that's coming out of, I think it's an RNA virus, is looking promising. But again, a lot of times things look promising until they get to the last phase, and then they just don't work. And so, like, I can't predict the future as much as anyone else can. All we can really do is keep doing what we individually can do to give other researchers the time they need to try to solve this problem.
So Daniel Jamu, who I just wanted to acknowledge, he's from IS. He was very helpful at setting this webinar up, so I wanted to acknowledge him here. He was actually asking about, there's so much talk about bats, and it's really not just about COVID-19. I think the SARS virus and then Ebola as well have bats as hosts. Is there anything that's special about bats? Why do we always see this?
Yeah, I'm so glad he asked that, because I asked this question myself yesterday when I was practicing my talk. That part of my own talk made me pause, and I was like, wait a minute. What about bats is so special? This is so weird. Why are they such a weird reservoir for viruses? And so I did a little bit of a deep dive into that too and started reading through that. It turns out that bats are a reservoir for a lot of viruses. It has something to do with how they are winged mammals. And so there's a tremendous amount of stress on the body when they are trying to fly. And you know, in any other normal mammal, it would trigger inflammation, it would trigger all these immune responses with that stress, the stress of flying through the air. And so they have developed a way, their immune systems have developed a way to sort of combat that stress. And in doing so, they have very measured and light responses to infections. And so viruses can kind of live in a bat without making them ill, because the bat immune system has ways to combat that as a side effect of being a winged mammal, which is totally bizarre. But hey, weirder things have happened in nature, honestly. So bats tend to be this weird reservoir for a lot of viruses, not just coronaviruses, but lots of others too, because of their biology. Just makes them good little disease factories, but they themselves don't really get that sick from those ones.
And Philip Cho is asking a question I think a lot of people have. Last week, there was some news reports about the WHO reporting there not being a substantial amount of asymptomatic spread of the virus. And he was just wondering, because then they sort of walked that back a little bit, if you had any thoughts about what...
Is this the report on asymptomatic carriers don't necessarily spread the virus, but pre-symptomatic people can spread the virus?
Yeah, and I think that's where a lot of the misunderstanding came from with respect to what was being said, but I don't know if you want to...
Yeah. So asymptomatic people are people who have probably a low viral load. They just don't have a lot of virus in them. And so therefore, they're not really having the same reaction that other people are having. And this is not unique to COVID. There are other illnesses where asymptomatic people can actually spread the causative agent. So the classic example is typhoid Mary. She was chronically infected with a salmonella bacteria, and she was a cook. And so what she would do is she would go and cook for people, and then everybody else would get salmonella except for her. And for the longest time, it just kind of went unnoticed, because why would she have it if she never was showing symptoms? And so that's like the classic case of the asymptomatic carrier. This person has it, doesn't realize it, and is going about their lives spreading it unintentionally. There was that study recently by the CDC saying that asymptomatic carriers of the virus will test positive for the virus, but are not likely to spread it compared to pre-symptomatic people, where again, the incubation time for this virus is like 2 days to 14 days. I think the average is 5 days. But people can still get sick up to 14 days later after becoming exposed. And so that probably means they were exposed to a very small amount of virus. And so it takes a lot longer for it to sort of build up to numbers in their system. And so before they become ill, they might be capable of spreading it around. Probably not nearly as much as somebody who is actually symptomatic, but can still spread it around. And they will eventually become sick. And so that's the difference. People who are asymptomatic never become sick. And these reports are saying that it's unlikely that they are contributing to the spread of the disease. It's more likely that people who don't realize that they are sick are going around living their lives, spreading it around, and then later become sick and only retroactively realize, oh no, I might have been spreading it, in that amount of time when I was pre-symptomatic.
So Yagana was asking, some countries are now starting on a second peak of COVID-19. Do you think a second wave is likely to happen this fall in the US before getting the vaccine?
This question's a bummer. So yeah, I mean, it's everywhere. It's everywhere. It's in every country. It's in every state. There are some states that are really rigorous about their social distancing guidelines, and the people are on board, and that's usually the states that were hit really hard during this first wave. So Massachusetts is on that list. You can't go anywhere without everyone wearing a mask. I mean, and it's just sort of, we're getting used to it now. And so social distancing measures start to ease and things start to open. People in Massachusetts are still more likely to continue wearing masks and continue taking those guidelines because it hit us so hard. But in other places where they weren't hit hard, that's not necessarily the case, and that those infection measures might go sort of overlooked. And so it is possible that we're going to get sort of this second wave. So we don't have a way to stop this illness right now. That's the bad news. The good news is we can slow it with social distancing and with rigorous testing. And so if we continue to increase our testing capacities, and so that when cases start to come up again, rather than shutting down everything, we can shut down certain communities or shut down certain states. And so we don't get the massive shutdown that we're getting, that we see now. And so hopefully, if things start to rise again, and they probably will, I mean, I would suspect they probably will come back and rise again, unless something dramatic happens to the biology of this thing where it becomes unstable. But we can't predict that, and we can't depend on that. Hopefully we can learn a little bit from this first pass and implement those as a second pass starts to rise, and then maybe the second one won't be so bad. But we got to learn from this. We got to learn from these experiences and learn from these mistakes that we make the first time around. Otherwise, we just keep repeating them over and over.
Thank you so much, Dr. Ellis. We really appreciate this. This was, I think, very informative for everybody. So
I hope so.
Yeah, and thank you everybody for coming, for the participation. That's really what we wanted to get out of this. And there was, almost too much participation, which I'm never going to complain about, because it's really.
If there are more questions that were unasked, always email me and I can send them to you, and you can give an update in the next one or whatever. Yeah. Thank you very much. Thank you for hosting this and for asking me to join. Yeah, I mean, I already did this deep dive, and having to do it again really forced me to go back and update. I had to update a lot of this stuff because it just changes so quickly. So yeah, practice those information literacy skills, guys, because we're going to need them.
Thanks, everyone.
Thank you.